Okay, so hopefully now you can see my screen. We're going to make a start now. So welcome everyone to our A Level AQA Biology session. In today's lesson, we are just going to be covering the structure of eukaryotic cells. Okay, so it was originally going to be a bit longer, but we are going to end a bit early today. But we were going to look at prokaryotic cells and exam practice, but I've just had to cut this short. So we're going to be looking at just the structure of eukaryotic cells in today's lessons. Okay, so this is a brand new topic. So we finished our work on nucleic acids, and now this is our next topic, which is all about cells. Okay, so this is what the spec says, and we are basically going to cover everything on the spec. We're just not going to come around to looking at exam questions. Okay, so we're still going to cover everything, but because we're covering it short today, we're not going to come on to some exam questions. Okay, so starting off, we're going to look at the structure of the nucleus. So this first lesson is all about looking at the structure of what we already have seen. We've looked at cells, we've looked at eukaryotic cells, we've looked at prokaryotic cells, we looked at them at GCSE, and we have looked at them at A level. But at A level, you're meant to sort of know a deeper knowledge of each of the structures. So at GCSE, we knew that there was a nucleus, we knew that there was cell walls, cell membranes, chloroplast, mitochondria, and so much more. Um, but over here, we need to know those structures and some of the internal structures of the organelles. Okay, and that's what we're going to basically look at in today's lesson. Okay, there are a few additional structures that we never covered at GCSE, but we will cover in today's lesson, such as the endoplasmic reticulum, Golgi apparatus, and so on. Okay, but hopefully by the end of today's lesson, we will look at all the structures that are present in eukaryotic cells. Okay, and we're going to start off by looking at the nucleus. Now, the first component of the nucleus that you are required to know is the nuclear envelope. Okay, and that's the double membrane, and it's the outer layer of the nucleus. So if we have a look at this image over here, this is our nucleus. This outer membrane over here is labeled as the nuclear envelope. What this is pointing at, this arrow is pointing at the nuclear envelope. Okay, so the nucleus has a double membrane, the outer layer of the nucleus, and that's called the nuclear envelope. The next point that we are looking about in the nucleus are the nuclear pores. Okay, now the nuclear pores are present in the nuclear envelope, and we've got an image, and I'll zoom in onto the image. But basically, this allows mRNA to come out the nucleus. We're not going to focus on mRNA because that's a later topic. But over here, as you can see, this the outer layer of the nucleus is this blue, or these blue lines, these blue bricked lines are the outer layer of the nucleus. Okay, and that's the nuclear envelope. Okay, so I'm just going to label that as blue. Okay, now the nuclear pores are basically the gaps that are present between the nuclear envelope. So as you can see, there are gaps present. There's a gap present here. There's a gap present here. There's many gaps. Okay, so you've got these gaps over here. And basically the gaps that you can see between the nuclear envelopes are what you call the nuclear pores. Okay, and the mRNA is very important in protein synthesis. Remember, the proteins are transcribed within the nucleus, and then they are transported out of the nucleus into the cytoplasm, where they attach onto a ribosome to synthesize the proteins. The sort of sequence of amino acids is generated within the nucleus, because the nucleus is the brain of the cell. It's where sort of the functions, it tells the cell what to do, all of that sort of stuff occurs in the nucleus. So the nucleus prepares those amino acids, the base sequence of amino acids, and then the base sequence of amino acids are then synthesized into proteins within the cytoplasm, which is outside the nucleus. So how is it meant to be synthesized in the nucleus and then transported out of the nucleus into the cytoplasm? Well, it happens through these nuclear pores. The outside membrane of the nucleus is the nuclear envelope, which is in blue as I've showed you on the diagram. The nuclear pores are the gaps between the nuclear envelopes, and those gaps are really important So the mRNA can leave from the nucleus into the cytoplasm and can attach onto a ribosome to start the process of protein synthesis. Okay, now if we have a look at the inside of the nucleus, we focus on the outside, we focus on the membrane and the gaps between the membrane. We also have the nuclear plasm. Okay, and the nuclear plasm is the granular jelly-like material. So in the nucleus, there is a fluid, as you can see in red, um, and that fluid is described as being the nuclear plasm. Okay, that's the jelly-like material, the granular jelly-like material which is present within the nucleus. Okay, and that's also highlighted on this diagram where this arrow is pointing to from the nuclear plasm. Okay, and then we've got the nucleolus, which is the final structure we're looking at in the nucleus. The nucleolus is this red circle over here. Okay, it's this red circle um, that is highlighted over here, the nucleolus, and it's basically where the rRNA is made. Okay, ribosomes are also made there, but it's basically where all the proteins are sort of synthesized. It's in the nucleolus of the nucleus. Okay, two very similar names. Okay, so the nucleolus is where rRNA is made. Again, mRNA, rRNA we come across later on in a later unit, so I'm not going to focus on them in too detail. But what you need to know are these four structures over here. Okay, the nuclear envelope, which is the outer membrane of the nucleus. We've got the nuclear pores, which are the gaps between the nuclear membranes, and that allows the transport of mRNA into and out of the nucleus, into the cytoplasm. You've got the granular jelly-like material, which is present within the nucleus, and that's called the nuclear plasm. And then you've got the nucleolus, which is the small circle which is inside, and that's where the proteins, amino acids, RNA, ribosomes are synthesized. Okay, so that's the structure of the nucleus. You also need to know the function, which you will already know. The function of the nucleus is that it's a site of DNA replication and transcription. Again, transcription just means making mRNA, which we come across later, but you already know that it's a site of DNA replication. And it also contains the genetic code for the cell. It acts as the brain of the cell. It tells the cell what to do. Okay, so that shouldn't be anything new. The point about transcription, it's where transcription happens. So basically, remember the mRNA is moving out of the cell and into the cytoplasm to synthesize proteins. Okay, now that mRNA has to be produced, and the process of making that mRNA is called transcription. Okay, so think about it. Your body has a lot of proteins. You have a lot of proteins in your blood. How are those proteins synthesized? How are enzymes synthesized? Remember, we looked at this when we looked at the primary, secondary, tertiary, and quaternary structure of enzymes. The primary structure, the sequence of amino acids first has to be produced. That is produced within the nucleus. The nucleus has the code to those sequences of amino acids. And once it's coded for the sequence of amino acids, the mRNA transports that sequence out of the nucleus. So then the amino acids can be joined onto each other. The correct amino acids can be joined onto each other via the ribosome. Okay, because the nucleus tells you what the code of amino acids is required. Okay, so that's all about the structure of the nucleus. We're now going to move on to the structure of the endoplasmic reticulum. Okay, now this is a new structure that you wouldn't have heard of at GCSE. But we'll take it step by step. Now, there are two types of endoplasmic reticulum. Okay, so you've got the rough endoplasmic reticulum, and then you've got the smooth endoplasmic reticulum. Okay, so we'll call this number one, and we'll call this number two. Now, there's a very easy way to remember the difference, which I will tell you. So both the rough and smooth endoplasmic reticulum have folded membranes called cisternae. Okay, now this is a really important point because first of all, we'll go through the structure of the endoplasmic reticulum because exam questions can ask you what is labeled or give a feature of the endoplasmic reticulum. So it's really important that you know the structure. And this is the most common structure that you will be asked to answer. You need to know that both the rough endoplasmic reticulum and the smooth endoplasmic reticulum, they have folded membranes. Okay, they have folded membranes, and these folded membranes are called cisternae. Okay, so over here, as you can see, we've got the rough endoplasmic reticulum, and it has these sort of wonky membranes. Over here, if I just like that, that's a wonky membrane. That's called the cisternae. And over here, the smooth endoplasmic reticulum also has that cisternae. It also has those wonky membranes. Okay, so that's what is meant by cisternae. Now, as you can see over here, the rough endoplasmic reticulum has these dots on them. Okay, there are dots present on these rough endoplasmic reticulum. And the smooth endoplasmic reticulum has no dots present on the smooth endoplasmic reticulum. Okay, as you can see, the surface is clean, whereas on the rough endoplasmic reticulum, we've got dots present. Okay, and that's the key differentiator. Those dots are known as ribosomes. Okay, and we know what ribosomes are. Ribosomes are responsible for protein synthesis, and that's the differentiator between the rough endoplasmic reticulum and the smooth endoplasmic reticulum. The rough endoplasmic reticulum contains ribosomes on the cisternae. Okay, so remember, cisternae are just the folded membrane. So as you can see, as I've highlighted on the diagram, cisternae are just the folded membranes. The ribosomes are present on the rough endoplasmic reticulum. So the dots are present on the rough endoplasmic reticulum are known as ribosomes. So if you ever have an exam question pointing to a rough endoplasmic reticulum, you must know that there must be dots if that is the rough endoplasmic reticulum. Okay? So the function of the rough endoplasmic reticulum is for protein synthesis. You probably guessed it. If it has ribosomes on the surface, then ribosomes are responsible for protein synthesis. So therefore, the rough endoplasmic reticulum is also responsible for protein synthesis. And in exam questions, what you will see is the fact that the rough endoplasmic reticulum has the dots. It has the dots, and on mock schemes, what you will say, if you sort of miss calculate the your answer, if you miss sort of if you write down the wrong answer by accident, if you write down ribosome instead of rough endoplasmic reticulum, or vice versa, the mock scheme will normally allow it because both the rough endoplasmic reticulum and the ribosome have very similar functions. But ribosomes are normally just represented by dots. Rough endoplasmic reticulums are normally have ribosomes on their cisternae. Okay, so the diagram for this will have dots representing the ribosomes on the outside. Okay, so we've looked at the rough endoplasmic reticulum. What about the smooth endoplasmic reticulum? The smooth endoplasmic reticulum is basically there to synthesize and store lipids and carbohydrates. So two very different functions, but the smooth endoplasmic reticulum doesn't have ribosomes, so it can't be involved in anything that is related to sort of protein synthesis or proteins or anything of that sort because it doesn't have ribosomes. Because it doesn't have ribosomes, it has a different function, and that function is to store lipids and carbohydrates. Okay, and the way that I remember this, we've got rough endoplasmic reticulum, or the RER, or for rough endoplasmic reticulum, and R for ribosomes. Okay, so rough endoplasmic reticulum has ribosomes, R for rough endoplasmic reticulum, and R for ribosomes. Okay, and then we've got the smooth endoplasmic reticulum, S for smooth endoplasmic reticulum, and S for store and synthesize. Yeah, and then you'll just have to remember the lipids or carbohydrates. Okay, so that's how I remember the key difference between the two. We've got the rough endoplasmic reticulum, which has an R for rough endoplasmic reticulum, R for ribosomes. Smooth endoplasmic reticulum, we've got the S for store, and S for synthesize of lipids and carbohydrates. Okay, so that was the endoplasmic reticulum. There are two that you need to know, the smooth endoplasmic reticulum and then the rough endoplasmic reticulum. Now, the next thing that we're going to move on to is the Golgi apparatus and vesicles. Okay, so this is also a new sort of concept. Um, so we've got the Golgi apparatus and vesicles. Okay, so again, the Golgi apparatus, can you see that it has these folded membranes? These are the folded membranes. Anything that has folded membranes are known to be cystine. Okay, and as you can see, we've got these sort of folded membranes. Okay, this is the Golgi, and we've seen these sort of folded membranes. Now, outside these folded membranes, we have these sort of circles coming off here. Okay, now you're probably wondering what are these circles? Well, these circles are what you call vesicles. Okay, these circles are vesicles. Okay, and they pinch off the cystine. So right outside the cystine, you've got these vesicles. Um, so I'll explain what vesicles do after we come on to the function. So the Golgi is where the proteins are processed, packaged, and modified. So remember, we've looked at the rough endoplasmic reticulum, which has a lot of ribosomes on it. And remember, ribosomes are there for protein synthesis. So that's where the proteins are sort of produced. Now, after the proteins have been produced, they're not like sort of ready to go straight away. They have to be processed and then they have to be packaged into their suitable sort of structure in order to be transported around the body. And that's what the Golgi apparatus does. So we've looked at the nucleus. The nucleus has the sequence, okay, the base code. So the ribosomes know which amino acids to join together. The rough endoplasmic reticulum has a lot of ribosomes, so the proteins can actually be synthesized. Once the proteins have been made, they have to be processed, they have to be packaged, and they can be modified. And that's what the Golgi apparatus does. It processes, packages, and modifies the proteins which have been synthesized by the ribosomes and the rough endoplasmic reticulum. Okay, and these are some other functions that the Golgi apparatus that can do. It can add carbohydrates to form to proteins to form glycoproteins. It can secrete carbohydrates. It can form lysosomes, which is our next structure that we're going to look at. It can transport, modify, and store lipids. So these are some of the. You don't need to know all of them. You just have to have a general gist of what they can do. Um, the function, the general function that you will be required to know is the Golgi apparatus is there to process, package, and modify the proteins which have come in from the ribosomes and the rough endoplasmic reticulum. Okay, these are some additional functions that you just have to have a general idea about. Okay. And then finally, once the Golgi apparatus has done processing, packaging, and modifying the protein which has come in from the ribosome and the rough endoplasmic reticulum, it will then transport that protein to the correct place through these vesicles. Okay, so once it's processed, packaged, modified the proteins, it will put the protein that's processed into one of these vesicles, and then these vesicles will be transported to the correct cell so they can fuse with the cell membrane and then release the contents. And we will look at this process in a later topic. It's called exocytosis and endocytosis, which we will come across later. All you need to know is that the Golgi apparatus will process, package, modify the proteins which have come in that have been synthesized. And then once it's done that, it will transport the proteins via the vesicle. It's called the Golgi vesicle to the correct place. Okay, so that's the Golgi apparatus and vesicles. Okay, moving on to lysosomes. Okay, now there's not much you need to know about the structure of lysosomes. You just need to appreciate the function. Okay, lysosomes are bags of digestive enzymes. Okay, they contain 50 different types of enzymes. Okay, and that's all you need to know. That's what lysosomes do. They have many different types of enzymes. They are bags of digestive enzymes, and then they can be released for many purposes. And here are some of the purposes that they can be used in. Lysosomes, they are contained within phagocytes. So you've looked at phagocytes at GCSE. We will look at them again when we look at the immune system. But phagocytes will basically hold these lysosomes. And if you get ill or you've got a pathogen which is coming in, the phagocytes will engulf the pathogen. Again, you've looked at that at GCSE. And how does it get rid of that pathogen? Well, the phagocytes contain lysozymes. Okay, lysosomes contain lysozymes. Okay, remember there's loads of enzymes present in the lysosome, and lysozyme is one of them. Okay, now lysozymes can hydrolyze the pathogen. Okay, they can digest the pathogen and hydrolyze the pathogen, so the pathogen is no longer present within the body. Okay, it can completely break down cells called autolysis. Auto means automatically, lysis means breakdown. Okay. Exocytosis, it can release enzymes to outside of cells to destroy materials. So again, if there's something that is not required within the body, the lysosome can release a specific enzyme outside of the cell. So then that enzyme can go and attack whatever shouldn't be there, and it can digest worn out organelles to reuse of materials. So that's another thing. It can release a specific enzyme that can digest worn out organelles that are no longer carrying out their function, so then they can be replaced into a more useful organelle. Okay, so that's lysosomes. The next thing we're looking at is mitochondria. Okay, now mitochondria we will look at again through a later unit, because you will look at photosynthesis and respiration. So we will look at chloroplasts and mitochondria again in more detail. But just to get you a general idea, mitochondria has a double membrane. Okay, its inner membrane is called cristae, which is folded. So the inner membrane is a folded membrane, and when you have a single folded membrane, it's called cristae. Cystony is when you have both the membranes which are folded sort of heavily. So we looked at cystony in the rough endoplasmic reticulum, in the smooth endoplasmic reticulum. Um, Over here, we're looking at cristae, the inner membrane, just the inner membrane of the mitochondria is folded, and that's cristae. Okay, inside the mitochondria, we've got the mitochondria matrix or the mitochondrial matrix, and that's literally the fluid center. So remember in the nucleus, we looked at the nuclear plasm, which was the granular jelly-like material which was present within the cell, and that's the fluid within the nucleus. In the mitochondria, we have the fluid center. There's fluid also present which is within the mitochondria, and we call that fluid the mitochondrial matrix. And then finally, in the mitochondria, you have loops of mitochondrial DNA. Okay, so if you have a look at this diagram over here, we have the inner membrane, which we looked at. We have the outer membrane. The inner membrane is folded, so it's called cristae. We have the mitochondrial DNA inside, and then we've got the intermembrane space matrix over there as well. Okay, and these are the cristae. The inner membrane is folded. We've got cristae. We've also got ribosomes present. Now we're coming to ribosomes on the next slide, but what you will realize is that we have normal ribosomes within our body, within our normal cells. Those are slightly larger than the ribosomes that are present in organelles. So you will see that the mitochondria and the chloroplasts, they have ribosomes present, but they have slightly shorter ribosomes present within the organelle, because obviously the organelle is a bit smaller, so you have a smaller sized ribosome present. Okay, and then you also have ATP synthase. We've already looked at ATP synthase. Again, we will look at it in more detail in a later topic, but you also have ATP synthase present there. Okay, and then finally, the function of mitochondria. The mitochondria is a site of aerobic respiration and ATP production. Okay, so we should know this definition over here. A site of aerobic respiration. You should have looked at that GCSE. And it also has loops of mitochondrial DNA. And the reason why it has that DNA is because DNA is needed to code for enzymes which are needed for respiration. Okay, remember for ATP synthase there, how is that ATP synthase going to be coded for? There has to be some sort of genetic material for the ATP synthase to be coded for. Okay, so that's the mitochondria. Okay, we're now going to move on to ribosomes. Okay, so again, you don't need to know about the structure of ribosomes because it's a really tiny structure. Um, but you do need to know what ribosomes are made of. And I've seen loads of exam questions ask this. Ribosomes are made of protein and or RNA. Learn this. Okay, because I've seen exam questions multiple times asking you what are ribosomes made of. Okay, they are made of a protein and or RNA. Okay, which is also known as ribosomal RNA. Okay, it is small and it's made up of two subunits, which is a protein and a raw RNA. There are two types of ribosomes. You have ATS ribosomes and then you have 70S ribosomes. Okay, ATS is the large ribosome which is found in eukaryotic cells. Okay, so remember, I was saying on the mitochondria slide that there's ribosomes present in mitochondria as well, but they're not going to be ATS ribosomes. They're going to be 70S ribosomes because they're not present within the cell, they're present within an organelle. 70S is the smaller ribosome that is found in prokaryotic cells along with the mitochondria and chloroplast. We're not looking at prokaryotic cells in today's lesson, but what you need to know is that 70S ribosomes, they're present in mitochondria and chloroplast along with prokaryotic cells, which we will look at in our next lesson. Okay, and then finally, the function of ribosomes. Ribosomes are a site of protein synthesis. Okay, so again, with ribosomes, there's not much new content. You need to learn this protein and or RNA it's made of. There are two types of ribosomes, ATS and 70S. ATS is the one which is found in eukaryotic cells, while 70S is the one which is smaller and found in prokaryotic cells because they are smaller, and mitochondria and chloroplasts because they are organelles. And we know from GCSE that ribosomes are a site of protein synthesis. Okay, with the vacuole, again, there's not much. There is just one key phrase that you need to know, one key word actually. First of all, the vacuole is fluid filled. Okay, so as you can see that there is cell sap present within the vacuole, and you should know that from GCSE. Now, the vacuole is surrounded by a single membrane, and you need to know the name of that single membrane. That single membrane is known as a tonoplast. If we have a look at this diagram over here, the tonoplast, it's surrounded by one single membrane, and in the specification it is named. If it's named, you need to know it. Okay, so it's known as a tonoplast. Okay, that's the single membrane which is present around a vacuole. Okay, so it makes the cell turgid and provides support. It doesn't have the normal cell wall. It has a special single membrane called a tonoplast, and it makes the cell turgid and provides support. The vacuole function, it's a temporary store of sugar and amino acids, um, as you know. And then the final point you won't really come across, but it's good to know, the pigments may color petals to attract pollinators. So again, it's present in plant cells. So that pigment can vary in vacuoles. And depending on the color of the pigment, it can color the petals of flowers, and that can attract different pollinators, different species. Okay, so the key takeaway from this slide is the fact about the tonoplast over here. That's what you need to know. And also the function which you should know from GCSE. Okay, chloroplast, same as the mitochondria, it's surrounded by a double membrane. We will come across chloroplasts again, and we're going to study the structure again later on in a later topic. But just for the purpose of today's lesson, I'm covering it. It's a site of photosynthesis, which is the same as it was at GCSE. Double membrane, same as the mitochondria. It's fluid filled. It contains enzymes for photosynthesis. That's all you need to know at the moment. And it contains thylakoids. Okay, these are folded membranes which are embedded with pigments. So over here, if we look at this structure over here, we've got the inner membrane, we've got the outer membrane. We saw that it was a double membrane. We've got the intermembrane space, which is just the space between the two membranes. These are thylakoids, okay? And the thylakoids, they are literally folded membranes within the pigment. So as you can see, these circles, they are folded membranes. Okay, these circles over here, each of those circles are known as a thylakoid membrane. Okay, and you can see that they come in stacks. Because they come in stacks, a stack of thylakoids is known as a granum. Okay, and these 2 points we will touch on later in a later topic when we look at photosynthesis. But at the moment, you just need to know that they contain thylakoids. Okay, and a stack of thylakoids is known as a granum. Okay, cell wall. None of this has changed. It's the exact same. This is literally everything you need to know. Cell wall is present in plant and fungi cells. In plants, it's made up of cellulose, which we've already looked at. In fungi, it's made of chitin. Again, from GCSE, you should know that. And its function is to provide structural strength to the cell withstanding osmotic pressure. Again, from GCSE, nothing has changed about the cell wall. Okay, we've already looked at the structure of the cell wall when we looked at biological molecules, so I'm not going to cover that in any more detail. Now, the final structure we're going to look at is the plasma membrane. There is a whole unit on the plasma membrane, which we are going to focus on. I'm just going to introduce it so you're aware that it's present, even though you should already know that it's present in eukaryotic cells. It's just the fancy word for cell membrane. So it's found in all cells because it's a cell membrane. A cell wall is there after the cell membrane. There is a phospholipid bilayer. Um, Again, there's a whole unit on this. Over here, you call the cell membrane the phospholipid bilayer. And all that means is that different molecules are embedded through the membrane on the outside. So as you can see, you've got cholesterol, you've got channel proteins, you've got carrier proteins, um, and basically you've got a lot of sort of phospholipids lined up against each other. Each of these, we've also already looked at phospholipids, um, but basically the purpose of the phospholipid bilayer is to allow different molecules to move across the membrane. It allows some molecules to move across, some molecules it is non-permeable to, it is permeable to. Some molecules have to move across channel proteins, some can go through carrier proteins, some can't move across at all. So it all depends on the phospholipid bilayer, the structures which are present. But again, this is something to cover later on because there is a whole unit on cell membranes. And finally, the function of the plasma membrane is that it controls the entrance and exit of molecules. Okay, so it controls what goes in and what comes out. Okay, and just remember that it's found in all cells. Okay, and those are all the structures that you need to know for eukaryotic cells. Okay, so we've looked at the cell membrane, the nucleus, the mitochondria, the chloroplast, the Golgi apparatus, Golgi vesicle, lysosomes, ribosomes, rough endoplasmic reticulum, smooth endoplasmic reticulum, cell wall, and cell vacuole. And that's all the ones which have been outlined by the specification.